Okay, hello everyone. My name is Clede Cumming, and I'm the Communications and Technology Manager at the Myotonic Dystrophy Foundation. Today, we're going to be going over the mechanistic aspects of the heart and CNS, part of the professionals track at the MDF Annual Conference. I'm delighted to introduce Dr. Thomas A. Cooper, MD. Dr. Cooper is a professor in the departments of pathology and immunology, molecular and cell biology, molecular physiology and biophysics at Baylor College of Medicine in Houston, Texas. After receiving his medical degree from Temple University Medical School, Dr. Cooper obtained his postdoctoral training at the University of California, San Francisco, where he initiated a long-standing investigation of alternative splicing regulation. He joined Baylor College of Medicine in 1989 and in 2003 became full professor and was named to the S. Donald Greenberg and R. Clarence and Irene H. Fulbright endowed chairs. His laboratory contributed to the identification of the RNA gain-of-function mechanism responsible for the molecular mechanism of myotonic dystrophy pathogenesis, specifically the disruption of developmentally regulated alternative splicing. His laboratory developed mouse models that inducibly express DMPK expanded CUG repeat RNA that produce DM1 disease features in heart and skeletal muscle. Currently, his laboratory uses these and additional experimental models to investigate the physiological roles of alternative splicing, as well as the mechanisms and consequences of its disruption in myotonic dystrophy. He will also be joined by Dr. Eric Wang, PhD. Dr. Eric Wang has always been interested in understanding how biological systems function, but in part entered this field because he has family members affected by myotonic dystrophy. He is personally motivated to understand DM and related diseases and to find treatments. Dr. Wang was an MDF fellow from 2012 to 2013 and was awarded an NIH Early Independence Award in 2013, which allowed him to start his lab at MIT. In 2015, he moved his lab to the University of Florida to join basic scientists and clinical researchers to study myotonic dystrophy and other repeat expansion diseases. Dr. Wang's lab is currently supported by grants from NIH, DoD, and the Chan Zuckerberg Initiative to better understand myotonic dystrophy pathogenesis and develop therapeutic strategies. With that, I'd like to welcome Dr. Cooper to the stage.
Thanks, Clede, and it's a pleasure to be here. I've been enjoying the meeting so far. Hope you are as well. Let's start with my share screen. There we go. Okay. So I have about 35 minutes worth of talking time. Knowing me, probably more like 35 to 40 minutes of talking time to leave time for questions and discussions afterward, which I certainly welcome. And I titled this talk, The Potential Mechanisms Causing Cardiac Features of Myotonic Dystrophy, because we don't really know. I didn't want to mislead, but there are some good leads. So here's an outline of what I'd like to talk about today. First, the first two items are to talk about the prevalence of cardiac features in myotonic dystrophy, and then talk about what those features are in particular. And the way I'm looking at this is to try to use this information to think about mechanism. What can we learn from the patients and from what they're experiencing in terms of trying to nail down where is the problem, in terms of where is it in the tissue and where is it in terms of in the molecular mechanisms as well. So then the second two items are defective conduction system mechanisms from mutation to cardiac. These two are, again, to try to look at mechanisms, dig deeper from tissue, from the symptoms and signs to the tissue, and then into the molecular features. What can we gain and what can we glean? And then the last few minutes is just to talk about some mouse models that are available, and particularly recently available to determine mechanisms and test therapies. So as we're all aware, myotonic dystrophy is a multisystemic disease affecting multiple tissues, as shown here on this diagram that I pulled off of the MDF website. And again, we will focus on the cardiovascular system, But I always find that since we tend to focus in our lab on the heart and muscle, it's important to remember that this is a multisystemic disease. And in particular, in terms of the mechanism, is the same mechanism happening in all those cell types? I think the chances are very good that yes, there is a common mechanism, but there also is high variability in this disease. Again, I think that we're aware of, and perhaps the modifiers, the genetic modifiers, the environmental modifiers, perhaps those will have some cell-specific or tissue-specific impact. So just something to be something to keep in mind. Have an open mind to the possibility of different mechanisms. The other factor that comes to mind is therapeutics. And again, if you had watched some of the recordings from the companies, and you see that they are thinking about the multisystemic nature. And while there does tend to be a focus on individual tissues, we do also need to think about hitting all those different cell types with therapeutics. And I think that will come up again when I talk about the different aspects of what potentially is going on in the heart. Okay, so let's talk about prevalence. There are a number of very good patient-reported outcome studies from different patient groups from around the world. I picked this one from the Christopher Project, mostly to show down here the abnormal, the prevalence of abnormal heart rhythm, but also compare that to some of the major symptoms: muscle weakness, fatigue, daytime sleepiness, the muscle and CNS symptoms, which affect a high percentage of DM1, DM2, congenital DM. And if you look at the impact score, how much does this impact the individuals? It's quite high. For heart rhythm abnormalities, what goes on with the heart? Again, half of the individuals are reporting that they have features of the heart disease. The impact score is less. And I think not that it doesn't bother individuals, but you can have arrhythmias, you can have a slow conductance within the heart and not know it. And so, but once there's an ECG, abnormality will be picked up. I think what's important about this is, I'm going to go to the second point here, that the cardiac features are the second leading cause of mortality. And the percentage of mortality ranges within the disease. I picked about a midpoint, about 25%. And that's due to sudden cardiac death due to lethal arrhythmia. So even though the individuals may not be noticing issues with heart, it's very important that they go to cardiologists and get the hearts checked out. I do have an MD after my name. I am not a clinician. What I'm going to be focusing on is the mechanisms, molecular biology, cell biology, mouse models. If I tried to see patients, I would be arrested, and as well I should. But luckily on Saturday at 2 p.m. Pacific time, there will be clinical updates to talk about cardiac management from a clinical perspective. So what are the main features, the predominant features of myotonic dystrophy heart when the heart involvement? It's predominantly electrophysiological. That is conduction delays and arrhythmias. And so I'm going to try to separate those to think about conduction delays through each heartbeat and also arrhythmias, and how the two are definitely connected, but how they also can be thought of separately. And so in particular, conduction delays, the time it takes for each heartbeat for the electrical signal to go through the heart as detected by ECGs, there's prolongation of the PR interval, the QRS complex, and the QT interval. And I'll be showing this in more detail in a moment. And again, there's a number of different arrhythmias, and particularly in the atria. And the main point I wanted to make here is that the main features that are, and particularly the life-threatening features, are electrophysiological. They are the arrhythmias, ECG abnormalities, but in particular the arrhythmias, and particularly the ventricular arrhythmias. When we think of heart disease in the general population, we think, of course, MIs, heart attacks, and heart failure. Heart failure is not that common in individuals with myotonic dystrophy. There are some decreased contractility detected by echocardiography, but that's not usually what takes individuals to the physicians. I think heart failure is probably less than 5% of the population. The predominant problem in the heart is arrhythmias and delayed conduction of the electrical signal. So again, with the idea that let's start thinking about what could mechanism be, how do we focus, where do we focus? Heart involvement in myotonic dystrophy type 2, not as well characterized, and there's also less known about the potential mechanisms. I'll get into the molecular biology of mechanisms in a bit, and it's very likely to be very similar, a loss of muscle-blind function. And there are arrhythmias and conduction delays that are observed, but appear to be less frequent. But then on the other hand, there are examples in which it was the cardiac manifestation of the disease that is what brought the individual to a physician and where DM2 was diagnosed. Okay, so this is thinking about the signal that goes through the heart. And again, I'd like to break this up into the idea of a slowed conduction through the heart and arrhythmia, and try to think about them separately, even though they are definitely connected. So with each heartbeat, the signal begins in the pacemaker, the sinoatrial node. That signal then goes through the atria, the right atrium into the left atrium. Then it goes through a conduction, modified cells that are conduction cells that move the signal to the apex of the heart. And over here on the right side, you can see what the conduction system looks like. So again, these are specialized cells that the Purkinje cells, a bundle of His, that carry the signal to the base of the heart. And what's illustrated here is you can see that the contraction starts at the base of the heart. So it can squeeze the blood, simply put, from the right ventricle into the lungs to get oxygenated, and from the left ventricle into the body for systemic delivery. And I do have a slide that shows that. So this is a mouse heart that's isolated. It's bathed in a fluorescent dye that is sensitive and fluoresces with depolarization. The heart is also paralyzed, so you're not going to see contractions. You're just going to be able to see the depolarization and repolarization. So here we started. You'll see a shot in the right atrium, left atrium, and then from the ventricles. But notice again, the signal comes down, comes from the apex and moves up. So, and again, that's the conduction system between the two ventricles that is carrying that electrical depolarization down to the base of the heart, and then it shoots up. So the contraction is starting from the apex to push out the blood. Okay, so with each contraction is the ECG. These are the different electrical signals that come from the heart from the surface ECG. Each of these waves have different names, P wave, QRS complex, so forth. And so what is measured is the distance from these different positions. A P wave to the R wave is the PR interval, and that is elongated typically in myotonic dystrophy. The QRS complex also typically takes longer through the myotonic dystrophy heart, and again, the QT interval. So the bottom line is that there is a delayed conduction with each heartbeat. There's a delayed conduction. What is life-threatening is a related but a separate feature is arrhythmias. So each heartbeat has this delayed conduction, but then there is also reasons to have a delayed or a disrupted rhythm. So the rhythm between each beat is typically equal between each beat. It can be too fast, which is a tachycardia. It can be too slow, which is a bradycardia. And I think even more striking is in this case, it's an irregular sinus block for different reasons. There could be the interval between beats can be different. And this is what becomes quite dangerous in terms of arrhythmias. So one question to think about is, where is the defect? Is the defect in the conduction system? Because there's heart block that takes place in myotonic dystrophy, or is it in the parenchyma? That is the communication from heart to heart. I mean, you saw in that depolarization and repolarization fluorescence, you can see that there must be connections from cardiomyocyte to cardiomyocyte. So the traveling of the signal through the heart between cardiomyocytes is important as well. So there was a study many years ago looking directly at the conduction system, the cardiac conduction system, in this case from 12 individuals with myotonic dystrophy, and using electron microscopy to look at what does the conduction system look like. And I've highlighted the two main things I'd like to point out. First is the most frequently observed histopathological lesions of the cardiac conduction system are fibrosis, fatty infiltration, and atrophy. And that is typically what happens in late stage disease of striated muscle, heart and skeletal muscle. But here's something that's important as well. The distribution and extent of conduction system lesions tends to correspond to the ECG abnormalities, including PR, interval problems, interventricular conduction delay, and bundle branch block in 4 individuals. And so there is a strong suggestion. There is a degeneration of the conduction system that is contributing to some of the features of the heart features of myotonic dystrophy, at least in DM1. But there also is disruption of the parenchyma of the heart. And again, just looking at the cardiomyocytes, and actually a cell type we're not talking about, which is a more more predominant cell type, is a cardiac fibroblast. But in this case, if we stick with the parenchyma of the heart as cardiomyocytes, there is also interstitial fibrosis, myofibrillar degeneration, abnormal mitochondria, again, myofiber disarray. And so this, in addition to the electrophysiological issues, potential of the conduction system, there's also potential heart damage, cardiomyopathy, that's responsible for the decreased contractility of the heart, the issues of contractility. Okay, so conduction system versus parenchyma, well, of course, that's too simplistic in biology. It's never either or. It's both, and then it's also the things that we haven't thought about. But in this case, there does seem to be a combination of both. You can imagine that the fatty infiltration, fibrosis within the heart tissue, that also will lead to arrhythmias, can lead to an irritation within a different place within the atria or the ventricles to set up an ectopic beat. Or you just need one cell to fire off strong enough to set off the coordinated contraction. Fatty infiltration and fibrosis of the conduction system. So the conduction system is involved well. And what I'm not talking about also are the gap junctions. There's not a lot of information, at least from human studies, on the gap junctions, which are directly responsible for the communication between the different cardiomyocytes. So again, going through this to think through the mechanistic possibilities. Where should we look? Also in terms of generating mouse models or different animal models, what cells should we target? Should we target the conduction system and the parenchyma? Also in terms of therapies, when we think about what cells to hit, multiple cell types to hit with therapeutics, should we be aware of, and I would think we should, of the conduction system of the parenchyma? Or in long-term disease, has the conduction system already degraded to a point? Is it going to be reversible? I think that's another question that we need to be thinking about. Could the therapeutics, if it is resolving issues in one component of the heart, could that be causing a problem with the other component not functioning? Okay, so that's from the tissue point of view. Let's go into some of the mechanisms. How do you get from this expanded repeat to disrupted electrophysiology? And I think most of you are familiar with the predominant mechanism of the disease, which is the expanded allele. It expresses the expanded repeat RNA that contains these tracks of CUG repeats. And a predominant mechanism is the sequestration of the muscle-blind paralogs in heart and skeletal muscle, paralogs 1 and 2. This was nicely shown by Maury Swanson's work and others. but the double knockout of these two paralogs very nicely reproduced very severe features of the disease. There is this counterintuitive upregulation of SELF1 that I believe is likely to be more involved in later stages of the disease. Both of these proteins are RNA binding proteins, which regulate these different RNA processing pathways, probably more so than what these arrows indicate, where the focus has been on disrupted developmental splicing and the regulation of alternative splicing. And what we as a field have learned, we've learned a lot actually from the study of myotonic dystrophy in terms of a normal process that takes place. And what we learned is that during postnatal development, right after birth, when tissues are undergoing this remarkable remodeling from a fetal function to an adult function, as expected, there's a lot of gene expression changes. There are also a large number, hundreds of genes that undergo these alternative splicing changes as well. And in terms of tissue remodeling, the example I like to use is imagine the muscle function of a newborn baby or even a 4-month-old compared to a 10-year-old, and think about how that tissue has had to remodel to become functional, and again, any tissue. So alternative splicing plays a role in this remodeling, and a large fraction of those hundreds of splicing changes results in expression of a different protein isoform, a transition from a fetal protein isoform to an adult protein isoform required for the adult function. And what happens in myotonic dystrophy, predominantly by splicing events that are regulated by muscleblind with some contributions of self proteins and perhaps some other proteins, but predominantly muscleblind. The disruption of muscleblind means that there's this subset of genes, I think a rough estimate could be 15–20% of those normally undergo this developmental transition do not. And so there's expression of the fetal isoform in adult tissues. That fetal isoform cannot fulfill the functions needed in adult tissues. And so that leads to symptoms of the disease. And I think other RNA processing, maybe some other events are also involved as well, but this is the best characterized. And the myotonia, with the failure to express the adult isoform of the muscle-specific chloride gene, is the best example, best proven example of this principle. Okay, so now we have hundreds of genes. Remember, we're trying to get to the mechanism of what's going on in the heart. Okay, so now I'm telling you there are hundreds of genes that are misregulated because of loss of muscleblind and perhaps contributions of self. How are we going to figure out which genes are involved? Is it that there are a little contribution from a large number of genes, and then how are we going to figure that out? Or could it be that there are large contributions from a small number of genes? And I'm going to give you my, it's either optimistic or wishful. I'm going to say it's optimistic. I think it is likely that there are going to be a relatively small number of genes that are misregulated at the level of alternative splicing that are likely to be causing the symptoms. And the reason I say that is because one of our experiences several years ago, in which we were looking at the regulation of alternative splicing during muscle differentiation in cell culture. We knocked out this RNA binding protein called RB-FOX. It inhibited the fusion of these were C2C12 myoblast cells that normally are proliferative. Then they differentiate, they fuse into those beautiful myotubes. They didn't fuse without this RNA binding protein. Hundreds of genes misregulated alternative splicing without RB-FOX, but it was a talented postdoc in the lab, Ravi Singh, who now has his own lab in Wisconsin, who was able to show that he was able to replace the correct isoform of 2 genes, and he was able to increase differentiation, the fusion, up to 50%. So we were able to at least get halfway to complete rescue using 2 genes, to the correct isoform of 2 genes. And I think that is likely to be a common phenomena. So again, we may not need to be looking when we correct a splicing event, don't look necessarily for 100% recovery, but look for some fraction of recovery. Okay, so let's get into the weeds. And now talking about genes and potential genes that could be affected. On the left is basically an action potential of a single heartbeat. And the reason I show this is basically to show the different genes and the different channels, ion channels, that could be involved. A sodium channel, a rapid influx of sodium, then an efflux of potassium, calcium channels, then all involved in not only in excitation, contraction, coupling, they're also involved in maintaining the membrane potential. And so here on the right are the different channels that are involved in, again, generating this action potential over and over again. And the main point here is that a large fraction of these genes undergo alternative splicing, and they are misregulated. A number of them are misregulated in heart tissue for myotonic dystrophy. So again, it's this idea of we can make a list of the genes that undergo misregulated alternative splicing, and how can we figure out which of these genes might be involved. And first we can do it by kind of thought experiments, but then at the end, I'll give an example of perhaps another way. So where I'll focus is on SCN5A, the sodium channel gene, where there is very good evidence for a role in the cardiac features of myotonic dystrophy. And what led to the identification of this gene was a few reports that came out, clinical reports that noticed that some of the types of arrhythmias in some individual myotonic dystrophy, DM1, looked like a Brugada syndrome, which is a certain type of arrhythmia. And mutations within the SCN5A gene caused the Brugada syndrome. And so then you look and you see that it is actually alternatively spliced. And then looking at splicing in DM, it was shown to be misregulated. So the focus is on these two mutually exclusive alternative exons. The fetal exon 6A is typically used in the fetal heart, although not 100%. This exon 6B is also, mRNAs containing this exon are also expressed. But there is a transition from the use of the fetal exon to complete use or almost complete use of the adult exon, and that's shown here by this RNA-seq data that comes from Nicolas Charlet-Bergerand's lab. And what you can see is this is RNA-seq data on unaffected hearts. And for RNA-seq, just think of thousands of sequence reads of mRNA from these individuals that are then piled up on the exons. Since it's mRNA, there are no introns. And so you can get a sense visually of the level of misregulated alternative splicing. So here's predominantly the adult exon is being used in unaffected individuals. Those who are affected, there's this increased expression of the fetal isoform. Does that have an impact? Well, the channel is actually quite interesting because it's a single molecule, a single protein that forms this channel. The alternatively spliced region is right around here, a transmembrane region, and part of this loop is actually one of the voltage sensing segments of the protein. The fetal isoform and the adult isoform, they actually only differ by 6 amino acids, but they do have different electrophysiological properties as detected by patch clamping. My simplistic understanding is that the fetal isoform is slower to open and slower to close during an action potential, so there's more of a rush in of sodium channels. What Nicolas Charlet-Bergerand was able to show was in a wild-type mice, he forced the expression. I should also say that this is conserved in mouse and human, that there is this transition from 6A to 6B, 6B being predominant in adult mice. He used AAV and a splicing redirecting approach to force expression, less expression of the 6B isoform, more expression of the 6A isoform, the fetal isoform, and he did show that there were ECG abnormalities consistent with myotonic dystrophy, and then also a propensity for arrhythmias consistent with myotonic dystrophy. This picture was taken when he was a student in my lab many, many years ago now. I think he looks about the same. Their lab also showed very nicely that this splicing switch is consistent with a loss of muscleblind, that this splicing regulation is sensitive to muscleblind, and loss of muscleblind is consistent with the use of 6A. So I'm very proud of Nicola, even though he scooped us a bit, because at the time, Paul Prang, a student at the time in my lab, he's now a postdoc in Joe Wu's lab at Stanford. We were working with a mouse in which we used line in which we used CRISPR to take out exon 6B from the germ line. And with that, you can then, you can make them a heterozygous. So one allele that expresses the adult isoform, one allele that does not. This is RT-PCR, so this band represents mRNAs that include 6A, the fetal isoform. Below is the isoform that's 6B. And you can see this transition from heterozygous mice, homozygous mice. And these mice also, consistent with what Nicola had shown, also showed propensity for arrhythmias, delayed conduction. And I wanted to show you this. If you remember the optical mapping of the wild type mice, this is what a homozygous animal that's only expressing the fetal isoform looks like. Right atrium, left atrium. And what you can see is a very delayed repolarization. So you can imagine that during this repolarization, if the atria was to fire off, if the SA node was to fire off while this was still repolarizing, you can imagine that could set up an arrhythmia, a circular electrical signal or circuit. Okay, other potential mechanisms from the literature in a mouse model from the Mahadevand lab, they showed that there's upregulation of the NKX 2.5 gene. Also, Nicola's lab showed misregulated processing of MIR1 microRNA, which is a very abundant microRNA expressed in heart that has a number of downstream targets that they showed were misregulated. And in more recent papers, there has been, and not too surprisingly, that calcium is going to be involved. Calcium is involved, as I said, in both maintaining the membrane potential, but also in excitation contraction coupling, the process of the voltage being sensed by the machinery within the cardiomyocyte, releasing intracellular calcium to cause the contraction, the sliding of the filaments. But there are several recent papers implicating a role for misregulated calcium. So again, perhaps calcium channels should also be on the docket to examine, and in fact they are. Okay, I wanted to end with talk about some mouse models relevant to heart. I know several of you have seen this slide already. I just think it's hysterical, so I'm showing it again. Okay, so this is a list of, well, I should also say there's a number of different types of animal models. What I'm focusing on is mouse models. I'm also focusing on the models that express the repeat RNA as something that's as close to what's happening in myotonic dystrophy, cardiomyocytes, and other tissues within the heart. There are also very nice models from loss of muscleblind that goes directly at the mechanism of loss of muscleblind activity, and also upregulation of SELF1. Again, and these models are the same with these models. We are generating phenocopies of the disease. I think our next step does need to be cause and effect, establishing cause and effect. And I also believe that these models will be very important for using as a mechanistic way to look for cause and effect. So there are just two that I will highlight that are more recent. One is from the Dirksen Thornton lab, And you can see the parameters here. It's expressing 250 to 400 pure CTG repeats. 80% of cardiomyocytes contain, or the nuclei contain foci. There's misregulated splicing. Also, importantly, there is this difference in the results from anesthetized versus awake animals. And in particular, when the animals are awake, there is a significant prolongation of the QTC and QRS, and not the PR interval. That's also true in the other mouse model as well. There may be something about mice. There is increased sensitivity for arrhythmias. The echo is unaffected, but that is, again, fairly consistent with a lot of what's going on in myotonic dystrophy. It's the electrophysiology that is a main feature. Again, not that contractility, we should not be thinking about that. And then there's some studies done on isolated cardiomyocytes, which yields very nice information. What I think is very interesting is that there was no change in the SCN5A splicing in these animals. So they had these electrophysiology issues, but no change of splicing in SCN5A. I'm going to end with the model that we just published, which again is very similar in what it has shown. We're expressing 960 interrupted repeats in the context of DMPK, exons 11 through 15. Again, there are foci with muscle-blind colocalization. We do see misregulated splicing, very robust, including SCN5A. We do see differences in conduction. About 10% of the animals during that 3 minutes of anesthetized ECGs, they do have arrhythmias, but with cardiac pacing, there is a significant increased susceptibility. I think an important feature is that when the repeats are turned off, because this is a TET-on system, there's reversal of the splicing changes, a reversal of the ECG abnormalities, and the susceptibility to arrhythmias. There's some changes with on echo. And if a student from my lab, Rongqi, who presented some data from this mouse model in her poster, if you got a chance to see that. And from RNA-seq data, certainly there was a lot of splicing changes, but we also did get hints of calcium handling abnormalities. All right, the summary in the future. I think really the main thing I'd like to point out is increased work with DM2. And again, it may be that I'm not up to speed on what's going on in DM2. So during the discussion, please jump in and tell us what you know. I think I'll just move to the acknowledgements. Thanks to the people in my lab, and in particular, thanks to the funding agencies, including the Myotonic Dystrophy Foundation, WIC, who have been supporting some of the work in our lab, as have different institutes from NIH and the MDA that are supporting either me directly or the individuals in my lab. And I thank you for your attention.
All right, well, thank you very much, Dr. Cooper. That was a very informative talk. I definitely learned a lot. You've been getting some shout outs in our chat here. I see John Fitzpatrick and Lisa Harvey-Duran. We're very happy to see you today. If you guys have any questions for Dr. Cooper, please just put them in the chat there, and I'm more than happy to read them aloud. I see we have one regarding the cardiac features in DM2. If there's any like similarities or differences you can talk about to DM1 expression.
Yes, again, I may not be the best to go in depth, and if there's someone online who can address that, that would be fantastic. My understanding is, my understanding is that there also are changes that happen over time, as at first it may be thought that in DM2 there isn't as strong cardiac abnormalities, but then I know that that has been increasing. It's become, you start to look more closely, and it's being found that there is cardiac abnormalities. Again, not as frequent, perhaps not as severe as in DM1.
Excellent. Well, thank you very much. That looks like all the questions we have right now. I
All
will
right.
invite you guys to please send Dr. Cooper a message if you have any questions after the session or later on during the conference. And after that, oh, I see we have one more that has come in. Why choose SCN5A? Why not a calcium channel?
Right. And I think because of the evidence that came from the patients, there was much more of an indication, the connection with Brugada syndrome, the fact that there were clearly kind of a slower kinetics and the action potential using the fetal isoform of SCN5A. The other thing I didn't get to is that in terms of deciding how much does one splicing event versus another affect and cause the phenotype, and I think that's where these mouse models come in. One of the things that we're doing is we're redirecting splicing of SCN5A in animals that are expressing the repeats to see whether that rescues the phenotype. And of course, with these mouse models, that's what you can do individually or maybe in combination. Redirect splicing. There are a number of ways to do that through AAV delivery of different moieties, and then redirect splicing, put it back to normal in the presence of the repeat RNA, and see whether how much does that rescue. Again, it's maybe not 100% rescue, maybe a 50% rescue.
Okay, well, thank you very much for that very detailed answer. And with that, I think we will bring Dr. Wang to the stage.
Great.
Thank you.
Hi, everybody. I'm going to go ahead and share my screen as well. That's showing up okay with the correct screen.
Looking great, thanks.
Great. Well, thank you, Clede, for coordinating everything, and thank you to the organizers of this meeting to give us the opportunity to share some of our thoughts and work around both the heart and the central nervous system. Tom did a great job going all the way from the sort of the phenotype, the symptoms that are experienced by patients, to the tissue level, and then to the cellular level, and then all the way to the molecular level. And I'm going to try to do something similar, although one of the notable aspects of the CNS is that we just don't know as much about how all of those different levels connect to each other in the context of myotonic dystrophy. The first thing I do want to emphasize, though, is that CNS issues are a really big obstacle faced by DM patients. So I'm just taking two of these word clouds from the Christopher project, which some of you may have heard a presentation about this project earlier in the meeting. Really amazing project in which information and feedback was gathered from both the patient community as well as caregivers. And I'm just showing a couple word clouds, really boiling down a lot of information about what some of the major obstacles are. And you can see that obviously mobility and muscle related symptoms are highly featured, but among the other really important obstacles include things that probably relate to CNS. So cognitive aspects, sleep issues, daytime sleepiness. And this is reported not only by the patients themselves, but also by caregivers. Later on in the Christopher project, there is a breakdown of looking at both at DM1, DM2, as well as congenital DM, looking at the top self-reported symptoms. And again, you can see that muscle issues or muscle weakness are highly featured, but in the context of all three, there are symptoms related probably to the CNS. So fatigue, while fatigue could be both muscular and CNS, but also learning difficulties, difficulty concentrating in CDM, and the daytime sleepiness, and others in the context of DM1 and DM2. CNS symptoms have been characterized somewhat extensively in the literature, although I think there's still a lot more to do, certainly in the context of developing outcome measures for therapeutic trials. But you can see here a range in a very nice review from Genevieve Gordon and Giovanni Meola looking at these different CNS symptoms, including, again, fatigue, sleepiness, but a number of features that may relate to executive functioning, also functioning of memory. So a lot more information certainly can be found in this review and others in the literature. I'll briefly touch on some work on imaging studies that have been done in DM1 in particular. There have been white matter lesions observed, overall brain atrophy or atrophy in certain regions. There has been ventricle enlargement that's been observed, and then also changes in volume in different regions. So overall reduced intracranial volume and volumes of certain regions of the frontal gray and white matter. Interestingly, there are certain brain regions that show increased volume, including the hippocampus and amygdala. Certainly a lot of work has been done in this area. I'm not the one that really is the expert at all in this area, but some of these references below, you'd be able to find much more detail on this. In terms of recognized molecular pathways in the context of CNS issues, one of the major pathways, of course, that has been first discovered in other tissues is muscle-blind sequestration. Self-hyperactivation has been not investigated as extensively as it has in muscle and heart, but it has been investigated to some extent. And there have been other pathways that have observed to be altered. So RAB3A upregulation, synapsin 1 hyperphosphorylation, tau aggregation. DM1 has been described as a peculiar tauopathy. And there have been characterizations both at the molecular as well as phenotypic level in mouse models in particular to observe potential alterations to both GABA and glutamate signaling pathways. So pathways that relate to overall excitation inhibition balance. There have been studies done primarily in mouse models that suggest that muscle-blind distribution may be shifted. This is probably related to muscle-blind sequestration. And this may have consequences for other pathways like RNA localization, which I really won't touch on too much today. It's important to note that, as Tom mentioned, there are other pathways that are characterized in other tissues, including muscle and heart. These other pathways just haven't been investigated as much in the context of the CNS, but they certainly could be playing roles in the CNS cell types as well. Briefly touch on some of the mouse models that have been developed and characterized to support studies of CNS issues in DM. And again, I will say that there have been a variety of models, but many models are being used in the context of muscle and heart, and the CNS efforts are slightly behind those contexts. I would say that the DMS-XL model is perhaps an exception. This is a model that was developed quite a long time ago and has been really an important model to reveal different CNS issues, at least in the context of a mouse model. So this model contains expanded CTG repeats, pure repeats in the context of human DMPK, and that's in a transgene. Tom's lab has developed a mouse model in which 960 repeats can be activated essentially by removal of polyacytes. And Guixin Wang has crossed that to a neuronal driver to observe changes in muscle-blind distribution and other phenotypes in the mouse model. And then Maury Swanson lab has recently developed, using CRISPR-based technologies, a knock-in model that contains 480 CTG repeats in the context of the mouse DMPK locus. There have been other models developed by Maury Swanson as well, in which the muscle-blind proteins have been depleted genetically, both muscle-blind 1 knockouts, muscle-blind 2 knockouts, and combinations in which both genes have been removed genetically through conditional strategies. So I'll spend the rest of the talk really focusing on some of our work to better understand CNS issues and basic pathomechanisms there. Of course, there's a lot of other work that's being done in the field, but for the purpose of this talk, I'm really going to focus on the work that we've done recently and published recently. And just to sort of frame these studies, we decided to look at frontal cortex, and why did we decide to do that? So firstly, as many of you know, CNS material, CNS tissue is not easy to obtain. The frontal cortex material from postmortem samples, it was a brain region for which we could readily obtain material and associated controls, age and sex match controls from different collaborators, including John Day, Takashi Kimura, and Charles Thornton, and the NIH Neurobiobank. The frontal cortex is thought to be associated with some symptoms that are reported in DM. And why look at the transcriptome? So characterizing the transcriptome in the context of myotonic dystrophy, we believe can guide studies about specific exons or genes that may drive different symptoms. In particular, because we know that alterations in splicing patterns play a major role, at least in other tissues like in muscle and heart. And some of that you've already heard from Tom. The transcriptome can also be a window in which you can inform biomarker discovery and mechanistic links to potential efficacy of therapeutics. So when we have therapeutic interventions, you'd like to have ways in which to measure how well that intervention is working, depending on the mechanism. And using the transcriptome can help guide what changes that one might expect. And as I mentioned before, the work that I'll mainly cover in the rest of the talk is from a recent publication from Brittany Otero, a graduate student in my lab. So before I get into describing our studies in the frontal cortex of the transcriptome, I think it's really important to frame exactly what are we dealing with when we get a piece of brain. The brain is a mixture of many different cell types. So there are neurons, there are astrocytes, there are microglia, there are endothelial cells, blood vessels running throughout, and those even are quite broad categories. So depending on the specific type of neuron, the particular RNA and proteins that are inside can be quite different. There are different regions of the brain that may have different properties and different expression levels of RNA and protein. And so, a lot of work has been done recently. The single cell RNA-seq field has really exploded to basically try to characterize a lot of these cell types. And so I just want to frame our studies in this way, because we are looking at all of these different cell types, essentially grinding them all up and trying to make some sense of what's happening. The subjects that we studied in particular were a mix of unaffected individuals, controls that we obtained from the NIH Neurobiobank, a range in ages of DM1 patients, and a small subset of DM2 patients. We performed Illumina RNA-seq, essentially what Tom described from that track of Nicolas Charlet's heart RNA-seq. We quantitated splicing patterns by a program called MESO, and quantitated gene expression by a program called Callisto. And to give you a sense for the kinds of data that we observe, if you look at different exons that we've identified to be alternatively spliced or differentially spliced in myotonic dystrophy, what I'm showing here on the left, the y-axis is this percent spliced in or psi value, basically how often is that exon used in the final mRNA. And different exons have different psi values at baseline. You can see in the light blue, the unaffected individuals for different exons start at different points, but every single one of these exons shown here shows a very different psi value or inclusion level in the myotonic dystrophy patients. Some of you may recognize these players. You have a potassium channel KCM-MA1, GABRG2, which is the gamma subunit of the GABA receptor. GRIP1 is a synaptic scaffolding protein, very important for helping to assemble other proteins at the synapse. And then you have other candidates on the bottom, CSNK1D, a major regulator of the circadian rhythm, MAPT, which is tau, and other synaptic scaffolding proteins. And so this is just a small handful of the genes and exons overall that we see are dysregulated. If you zoom out and look more globally and take the average of the splicing values for your unaffected individuals and compare that to your DM1 individuals, you can find a lot of different candidates. And from this, we essentially identified about 130 high confidence misspliced exons. What you'll appreciate from the graph on the left is that the spread of the data in the DM1 individuals tends to be larger than the spread in the unaffected individuals. So why is that, right? Basically the extent of missplicing is variable. And one of the ways that we think about myotonic dystrophy, it's a repeat expansion disease. So it's not really, the way that we think about it, it's not really that either one has the mutation or one doesn't have the mutation. We think of the mutation as more of like a dimmer switch, where you might have a repeat that could be longer or shorter, and that actually creates differences and variability in the downstream molecular changes, as well as the phenotypes that you see in people. We've observed a wide range of splicing severity across patients in muscle and heart. So on the left is RNA-seq data from a variety of tibialis anterior biopsies collected by Charles Thornton's group. Each column is a different individual and each row is a different exon. What you can appreciate is that there's this gradient, sort of this triangle that comes down in red, where the events at the top are misspliced only in a subset of patients, but some of the events at the bottom are misspliced in most of the patients. You see that effect also in the heart, and the gradient is a little bit harder to see because we don't have as many samples, but you get the same sense. So we wondered whether we see the same thing in cortex. And one difference between the cortex versus the muscle, of course, is that the cortex is postmortem. The heart data here is postmortem as well. But we indeed did see the same sort of gradient, where splicing events on the bottom tend to be affected in almost every DM1 patient, whereas the events at the top were maybe more mildly affected in a subset of patients. In the past, we've been able to take subsets of events and essentially take their average and derive a total dysregulation score, total splicing dysregulation score, and you can see that up top. The patients are essentially ranked by that order, and that's why you see this gradient. So what's really driving the differences in severity of missplicing? In some of the previous talks today, the repeat instability session, there was a lot of discussion about somatic instability. And I'm showing a Southern blot from Charles Thornton from a number of years ago now, where he made the observation that in the same individual, if you look at the repeat length here by Southern blot, where you basically take the DNA, you run it on a gel so it separates by size, and you transfer it and you probe for the repeat containing DNA. In blood, the repeats are much shorter than what you see in muscle. So it suggests that the repeat length is actually changing over time in the affected individual, and that it could be quite different in different tissues. Somatic instability is very important, not only in myotonic dystrophy, but in a lot of other repeat expansion diseases. But one of the challenges has been to definitively show that the repeat lengths actually correlate to molecular phenotypes or even phenotypes in people. Generally, there's been a very good correlation in age of onset to repeat length. Individuals like Darren Monkton and Fernando Morales have done really nice work to show that if you estimate the progenitor allele length, you can get really nice correlations. But it's been generally difficult to correlate to molecular phenotypes. And the other question that we've had over the years is, is it that certain cell types or certain subsets of cells, do they expand faster or slower than others? If certain tissues expand faster than others, then you would expect that might be the case. But we just don't have a good sense for that. So you could have a situation where you might get modest expansion across many different cells, or perhaps it's extreme expansion in fewer cells. And it sort of changes the way you think about what's actually happening in the tissue. We wanted to measure the repeat sizes in some of these samples, and it generally is challenging with samples in which you have limited material. Southern blot has been a gold standard, although it's somewhat low resolution in the sense that you get a smear, and it does require enough material to be able to do the assay. There have been assays like small pool PCR where you can PCR across the repeats, but it can be quite challenging for extremely long repeats. And so a direct measurement would be ideal. For example, if you can imagine just stretching out the DNA labeling it and measuring the distance between those two labels such that the repeat is in between, you might actually be able to directly visualize and measure that DNA. And we know that if you stretch out DNA linearly, 1 kilobase is about 500 nanometers, and that's in the range of what one can resolve using optical microscopy. And so we took advantage of a particular technology that is called optical mapping. It uses DNA nanochannels. What you can see here is that DNA fragments are being flowed through this flow cell. And here you can see they're actually getting stretched into individual double-strand molecules. And the machine, the instrument that we use, the BioNano Sapphire, essentially allows you to take pictures of that DNA with the labels on it. You know where the labels map based on the particular sequences that they label, so the sites are predetermined. And you can find the fragment that maps to your particular locus because you know what pattern your labels might yield. And so this is an example of some of the data that you get from this kind of assay. So the particular DNA fragment that we're interested in has these, particular markers. These 4 labels there, they flank the CTG repeat with known distances, so we can identify that fragment in that sea of DNA molecules. If you look at an unaffected cortex, a control subject, and you calculate the distance between the markers to estimate number of repeats, you get this very tight distribution. There is some error in the technology and the measurement just based on the microscopy, and that's why it's not exactly a single number. But you can appreciate it's quite narrow there, and you can infer a percentile. So at the 50th percentile, your estimated repeat length is basically close to 0, or maybe 10 or 20. But in the context of a DM1 cortex, the actual reads are on the left. You can now appreciate that about half the reads show that unaffected repeat length, but then about half of them show a very extreme longer repeat. And again, on the right, you can calculate, show that in a histogram format. The blue line is a cumulative distribution where you can estimate sort of the 50th percentile. Again, it's close to 0. But if you look at the 75th percentile of lengths, you're around 2,000. The 90th percentile, you're around between 3,000 and 4,000 CTG repeats. So that's an example of the kind of data that you get. We decided to profile a number of the samples for which we had splicing severity measurements. And so here they are ranked in order of splicing severity based on our RNA-seq data. We decided to analyze the individuals where the arrows are. And when we did that, and we started to assess the repeat length, we saw something that's quite interesting. So when you take the 50th percentile of repeat lengths, this would not be expected to correlate with anything really, because it's just the threshold between your normal length allele and the expanded allele. You really don't get a correlation with splicing severity. But if you correlate to the 70th percentile, you start to see a correlation emerge. And if you go to 90th percentile, the correlation between the length of the CTG repeat and the extent of missplicing is actually quite striking. It's quite good. So there's a couple different ways to interpret this, and we don't know which answer really is correct yet. I think it requires some more detailed analysis. But one idea would be you perhaps have a particular subset of cells that are really expanding quite dramatically, that contribute a great deal to the splicing dysregulation. Another hypothesis would be that you have a specific cell type. Let's say it's the neurons. The neurons might be the ones that expand the most, and they're the ones that are contributing the most to the splicing severity. That's a possibility. To really nail that down, we'd have to isolate specific cell type and measure the repeats there. And then a third one is simply that this metric is the most accurate way to assess sort of overall. This metric is the best way to assess repeat length, and that's why you get the best correlation with missplicing. So we don't quite know the answer as to why only looking at the 90th percentile gives you the best correlation. But there's ongoing studies to really investigate that. Okay, so if the repeat lengths really do correlate with missplicing, then what's in between the repeat length and the missplicing? So if you look at individual splicing events here, you do see that their dysregulation correlates with each other, right? So the missplicing of GABARG2 exon 10 correlates with the missplicing of MDL2 exon 5, and also between DL-GAP1 and GRIP1 exon 21. So there's some shared driver. We know it's the repeats, but what's directly downstream of that? When we analyze the high confidence exons and look at the motifs that are around those exons in the intronic space, we find that you get an enrichment of certain types of motifs. These motifs tend to be enriched for muscle-blind binding sites, which are pyrimidine, GC, pyrimidine. And you find them in the expected locations where the exons that are aberrantly included tend to have MBNL motifs upstream, and then the exons that tend to be excluded tend to have the motifs downstream. There are some other motifs that pop up, and we don't know exactly what they bind or what RBPs might regulate them. Those would be the subject of future investigations. So given that muscleblind is really has a signature for a lot of the exons, one question one might ask is, to what extent is muscleblind depleted? This has implications for understanding therapeutic strategies. How much muscleblind would one have to replace, or how much muscleblind would one have to release from repeats to get rescue of missplicing at least, and perhaps symptoms. So we took advantage of the mouse models that Maury Swanson has generated. He's generated muscleblind 2 knockouts, as well as 1-2 double knockouts in neurons. And there's been other models, but these were the ones we focused on. And we decided to look at a subset of exons that are alternately spliced in both mouse and human and dysregulated in these systems. So we were able to use 77 human mouse exon pairs, and we essentially ranked the muscleblind knockouts among the wild type mice in terms of splicing severity. So to the right is the most severe, to the left is sort of unaffected. So the wild type mice are on the left, and then the double knockouts are all the way on the right. We were able to use those human mouse exon pairs to compute the same score and basically put human and mouse on the same axis. And so the unaffected control individuals, they all cluster on the left. But then you see that most of the patients at postmortem essentially have an intermediate state between a 2 knockout and a double 1-2 knockout. When you look at the expression level, at least at the RNA level, and you try to place where human DM1 brain lies, we estimate that about 66% of muscleblind is functionally depleted in most of the samples. Essentially all of them except that one very mildly affected human on the left. The next question we were really interested in was, what's the overlap between other tissues in terms of missplicing? So we do believe that at least a major driver of missplicing in the different tissues in muscle, heart, and frontal cortex is driven by muscleblind. So you might expect a lot of the same shared targets. When you do that overlap, that Venn diagram, there's actually surprisingly little overlap between the tissues. You can see that there are overlaps between any given pair of tissues that's reasonably large, but there's not that much overlap among all three. Three of the best, most convincing events that are shared in dysregulation across the three tissues are shown here: muscleblind 1 exon 5, muscleblind 2 exon 5, and tau exon 3. So from one standpoint, this is somewhat surprising, but from another standpoint, this could be potentially useful for biomarkers where you might be able to unambiguously assign therapeutic efficacy to a particular tissue that you're trying to target. If you look at pairs of tissues, if you look at the frontal cortex versus the TA, or the frontal cortex versus the heart, and you look at the dysregulation, the shared targets tend to be dysregulated in the same direction. So the environment of the cell is changing in the same way. It's just that the specific targets might be a little bit different in each system because of differences in the way they're regulated in each tissue. We did do a little bit of sequencing on DM2. It wasn't as extensive as DM1 just because we couldn't get access to as many samples. But we did analyze those samples as well. And we did an overlap between DM1 versus DM2 regulated exons. We didn't have the same number of samples, so we had to be careful with this analysis. We did some subsampling approaches to try to get around the issue of power here. But when we do that, we see that about 30% of the exons misspliced in DM2 are also misspliced in DM1. You can see a scatter plot on the right. Basically, you have a lot of exons that are sort of trending towards dysregulation in DM1 or DM2 or vice versa, but not quite there. So why is the overlap not larger? There have been studies from Nicolas Charlet that suggest that there are other RNA binding proteins that play a role in DM2. So one of these RNA binding proteins is RB-FOX, the RB-FOX family. And RB-FOX has been thought to be able to compete with muscleblind to bind CCUG repeats in DM2. And you can appreciate from the canonical binding motif of RB-FOX, UGC-AUG, that actually looks quite similar to CCUG. It's essentially the same motif, except the A is now a C. There have been other, so one of the mysteries that we came upon was that when we did motif analysis on the DM2 regulated exons, we didn't see an obvious enrichment of those particular RB-FOX motifs. The UGC-AUG really didn't pop up. But there have been recent studies from Chris Burge's lab, my PhD mentor, where they found that at high concentrations, RB-FOX may actually bind to intermediate affinity sites to regulate splicing. So these are sites that don't look like UGC-AUG, but they're still bound by RB-FOX when RB-FOX concentrations are really high. So if you look at the concentration or expression level of RB-FOX relative to CNBP, the gene that contains the CCUG repeats, or you look at RB-FOX relative to MBNL in frontal cortex or TA versus TA, RB-FOX is really high. Total RB-FOX is really high in the frontal cortex versus the TA. And then when you do motif analysis on the DM2 exons, you find these motifs that we initially did not recognize, but many of them tend to be those motifs that are the intermediate affinity RB-FOX motifs. The plot on the right, the spine and seek enrichment, essentially is a measure of how well RB-FOX binds to that motif when in high concentration. And so perhaps what's happening in frontal cortex is that you're depleting from a very, very high level of RB-FOX to something that's somewhat intermediate. And so you don't see the obvious RB-FOX motifs, but you see some of the weaker motifs that are still playing a role. We also looked at gene expression changes in the frontal cortex samples. And what we noticed was that genes that are upregulated in the DM1 cortex tend to be enriched in inflammatory pathways, and genes downregulated tend to be enriched in neuronal pathways. Of course, anytime that you see something that looks like this, downregulation of neuronal genes, upregulation of inflammatory pathways, you wonder, is this simply a shift in the cell type composition? As I mentioned earlier, there have been a number of studies that show white matter and gray matter alterations, changes in brain volume, changes perhaps in the composition of cell types. So we wanted to accurately assess this. We ended up having to develop some methods to estimate the proportion of different cell types from bulk RNA-seq. So essentially, if you get a mixture, everything's ground up. We used existing single-cell RNA-seq data sets to try to recover the proportions of the different cell types that went into the mixture. We first used an Alzheimer's disease entorhinal cortex data set to confirm our method. In this data set, we knew that the contribution of neurons decreased, and then the contribution of microglia and endothelial cells increased. And we just wanted to make sure that our method worked properly. So we took that method and we applied it to our data set. And what we see is that there actually isn't any sort of obvious shift in cell type composition, at least in neurons, endothelial cells, oligodendrocytes, or astrocytes. We did see a slight shift in increase in microglia. So from this, we think that the neuronal downregulation is likely not due to a change in the composition of the mixture, but it could simply be that the neuronal programs actually are being downregulated within those cells. After having deconvoluted cell type contributions, we wanted to know whether missplicing events occurred in many different cell types as well. On the left, as I mentioned before, the left is the gene expression changes. You can see there's a huge enrichment in downregulated genes in the neurons, a huge enrichment in the endothelial cells and the microglia and upregulated genes. But in the middle, you can see that among the genes that either show no change in splicing or the genes that show changes in missplicing, it's kind of across the board. Many different cell types show these missplicing signatures. To truly nail this down, we obviously will need to go to a higher resolution and actually assay those specific cell types. But at least preliminarily, that's what our data seems to suggest. So just as sort of a last analysis that we did, we wanted to tie this back to ways in which we could harness this information for potential therapeutic development. First, we wanted to know, generally, does splicing correlate with gene expression? On the x-axis is the extent of missplicing, and on the y-axis is the extent of gene expression dysregulation. And you can appreciate that actually those two metrics correlate quite well. Why did we want to do this? We wanted to know whether gene expression might be a useful readout, because gene expression might be easier to assay in, for example, cerebrospinal fluid. So on the right, we took a existing data set of cerebrospinal fluid proteomics, and we overlapped our data set. On the x-axis are basically a metric of how well the gene correlates with missplicing. So the farther to the right it is, the better of a biomarker it is that relates to underlying missplicing. On the left, on the y-axis is just the overall expression level. And then what's highlighted are those proteins that have been able to be identified by proteomic studies. So the idea is that perhaps some of these candidates might change in their abundance in response to disease status in myotonic dystrophy. So I'll just wrap up to sort of summarize some of the implications for therapeutic approaches and outcome measurements in the context of the CNS. So I think that something that was somewhat surprising to us was that all individuals studied have at least 1,000 CTG repeats. There was one individual who had very, very mild splicing defects, but they still had quite long repeats. So this could mean that you actually need quite long repeats and need a lot of somatic instability to get phenotypes that could bode well for approaches that impede somatic expansion. It could also bode well for approaches that take advantage of the immensely large repeats, advantage of taking advantage of that stoichiometry. We know that muscle line depletion can account for many of the missplicing changes, so that does seem to be a useful therapeutic pathway or therapeutic target. And I think that one of the notable findings that's consistent with other observations in the literature is that some of these missplicing events might affect excitation inhibition balance. There are a lot of therapeutic approaches out there that actually target that particular axis, either GABA signaling or NMDA receptor signaling. And so those are pathways that may be important to look at in the context of therapeutics. We have seen that multiple cell types are probably affected in DM1, but it still remains to be clarified which cell types are the most important to rescue or to target. Of course, it would be great to target all of them, but depending on the particular disease modality, you'd really like to know which particular target you need to go after to have an impact. And finally, biofluids may contain biomarkers that arise from these alterations. And it's our belief that it's important to link the mechanisms of how those biomarkers arise in order to be able to interpret shifts in potential therapeutic trials. And so with that, I'll thank a number of the individuals that did the work. Most of that work was done by Brittany Otero in collaboration with John Day, Takashi Kimura, and Charles Thornton. Kairo Pukalov, another graduate student in the lab, led the repeat sizing efforts. And a lot of this work was supported by MDA and NIH. I'm happy to take questions.
All right, thanks for a great presentation. Really enjoyed the graphics and visualizations you had included, as well as all the other people in our ecosystem you referenced. So it looks like we have a couple questions here for you that are pretty detailed. So the first is, according to your observation, there is a variation in repeat numbers in different tissues in DM1 patients. How will you count for the severity of the disease? Which tissue will you take into consideration?
Yeah, this is very interesting and an important question. I don't have an answer to your question. I think that generally we look at repeat lengths in blood. I think there are good ways to estimate sort of the progenitor allele length. For example, Darren Monkton and Fernando Morales have done a lot of this work. But you're right in the sense that you could have individuals who have different disease severities in different tissues at different rates. And I don't have a good answer for you in terms of how to deal with that. Something that is somewhat reassuring is that I believe some work from the DMCRN has been looking at correlating metrics in different tissues. And I know that, for example, some of the functionality in something related to grip strength seems actually to correlate quite well with differences in biomarkers in tibialis anterior, looking at splicing there. I could be slightly off on the details of this, but the idea is that even looking at different muscle groups, that there are some reasonable correlations. Now, between tissues, I think it's still an open question.
Excellent, thank you. It seems like we have a couple other kind of related questions that I'll go over. We have two from Dr. Matthew Disney. He asks, the overlap in splicing is interesting. Does each outcome better correlate with expression? That is, are pre-mRNAs similarly expressed in all tissues similarly affected in splicing alterations in DM1 tissue?
Yeah, so that's interesting. So it's not just that the differences in the different tissues are present because the genes are not expressed. So there are some genes that are expressed in both frontal cortex and TA, but the dysregulation only occurs in one or the other. And I think that the explanation for that is that there are probably other RNA binding proteins in each tissue that can compensate for the loss of MBNL. So the dose response curve of the concentration of MBNL that's important for regulating a splicing event is just different in different environments. I think that addresses your question. There's a lot of examples of that when you look at different pairs of tissues.
Excellent. And then he had a kind of follow-up comment question is, you suggested that different cells can have different somatic expansion. Do you know if these cells have gene expression differences that could rationalize this, i.e. DNA, repair protein expression differences?
Yeah, and I'm definitely not the expert on this, but for example, MSH3 is one of the major players here, and certainly there are SNPs that have been found in MSH3 that mediate differences in instability expansion rate, and certainly MSH3, for example, if you knock it down or overexpress it, that certainly impacts rate of somatic instability.
Excellent. Well, that looks like all the questions we have today. Thanks so much for a great presentation. We do have one more that just came in, actually. The low splicing change in 50% percentile, do you know if the levels of MBNL1 or MBNL2 proteins in different cell types to make stoichiometry predictions, numbers of protein and RNA molecules with different repeat length?
Yeah, so Tom, I think Tom, you're asking about the number of protein.
Jump back on Tom if you'd like as well.
Yeah, you want to?
Yeah, it makes it much easier. Yeah, I guess just the idea that I was so surprised about the 50th percentile. You have these long repeats, but there still isn't that much of a splicing change. So, I mean, is it because there's a lot of muscleblind being expressed in those cells?
I should clarify, Tom. The 50th percentile is simply the cutoff between the unaffected allele and the expanded allele. So that's sort of, you wouldn't expect a correlation at that point. So it's a little confusing. The 90th percentile really is sort of the the 80th percentile of the expanded alleles. I should sort of say that.
I got it. Okay.
But you're bigger. But I think the bigger question
Yeah,
is
but the idea that you have
Yeah.
1,000 repeats
Yeah.
with less splicing change.
that's right.
Just
And
maybe there's more muscle blind in those cells, a higher level of muscle blind in those cells.
I think you're right in the sense that there's this balance. It's not about the total repeat load or the total MBNL load. It's really about the combination of both. It's about their ratio.
Yep.
And then what is the set point for that ratio? I think it's actually quite long. And you know, in the lab, it's really challenging to make long repeats. So we have to resort to expressing more of them, not longer of them, right? And that's really been what's more successful in the animal models. But part of you wonders, well, is there something different about how we're modeling the disease that is not truly reflective of what's happening in the patient? And so I think that's an important point we all need to keep in mind.
Excellent. Well, thanks for adding in that last little question. It was great to get a little back and forth between you two. And thanks so much to both of you for a great session. I'm going to invite everyone to go to our special performance by singer-songwriter Eric Hutchinson that's about to begin. And I hope you all enjoy the rest of the MDF conference.